Hello, my name is Dr. Dewan S. Raja. Today I will discuss about the histology of the mitochondria. Mitochondria are the organelles. They are double membrane bound structure like other organelles, contain circular strand of DNA that is different from that of the DNA of the nucleus. Here it is circular, and nuclear DNA is strands are not circular, they are elongated. Synthesis most ATP during aerobic cellular respiration by digestion of fuel molecule like that of from carbohydrate will get the pyruvate that will be converted into acetyl coenzyme A, then from fat will get by beta oxidation acetyl coenzyme A, and that will be ultimately converted into ATP by the Krebs cycle. Okay, so if we look at the structure of the mitochondria, we have the outer membrane, inner membrane, intermembrane space. We have the shelf-like projection that is called Cristae. The matrix here, the matrix is the intermembranous space here in intermembranous space, and this is the electron microscopic picture of the mitochondria. Okay, if you go there, then so number and location of the mitochondria is variable. It is increased where more energy is demanded, and mitochondria can move from one part of the cell to another part of the cell. So it is like a movable power plant. Okay, so its number is high in the cells where more metabolism, more activity like cardiac muscle cell, skeletal muscle cell, and the epithelial cell of the proximal convoluted tubule. In the neuron, it will be very high in number. In the hepatocyte, maybe around 2000 mitochondria in a cell, but cell number is decreased where less activity, like in the osteocyte cell or in the adipose cell will get less number of mitochondria. A mitochondria has outer and inner membrane. Inner membrane include enzyme assemblies of electron transport system and ATP synthesis. So this is the inner membrane. We have that is the site of ATP synthesis, like that area, and we have this is the ATP synthetic complex. ATP synthetic complex, these are for the ion transfer, this part. Okay, so mitochondria, here is the hepatocyte. There is the parenchymal cell of the liver, and we are getting mitochondria, multiple mitochondria here, maybe around 2000, even more. We have also other organelles like that of the endoplasmic reticulum. So mitochondria is present in the cytoplasm like other organelles, and a lot of other cells around the hepatocyte we are seeing they have also mitochondria. Mitochondria is only absent in the red blood cell or erythrocyte or in the terminal part or the most superficial part or layer of the keratinized stratified squamous epithelium. We call them keratinocyte. Cytochrome C from the inner membrane that is present in the intermembranous space, okay, that actually involved in apoptosis. In stress cell, apoptosis is the programmed cell death without inflammation and without pain. Okay, so mitochondria have their own DNA apart from oxidative phosphorylation and produces ATP. It is the adenosine triphosphate. It is a stored form of energy. It can synthesize lipid. That lipid make its membrane. Especially, we have to know that one of the lipid, phospholipid called cardiophyllin, may make the inner membrane more or less impermeable or close to impermeable. Can produce heat and maintain body temperature. Some of the energy is disseminated to our body and maintain our body temperature. And the mitochondria of the brown fat cell in the hibernating animal, they produce heat also, specifically for the hibernating animal, but we also get heat from mitochondria. So not only necessarily always the ATP and also the heat that is transmitted to our entire body and maintain body temperature. Mitochondria take part in apoptosis by means of cytochrome C. We discussed that part. Okay. So origin and replication of mitochondria. Mitochondria evolves from aerobic bacteria. Mitochondria enters the cell. So cell is the eukaryotic cell, or you can say eukaryotic anaerobic cell. It enters the cell in the process of evolution and symbiosis. So mitochondria get protection and nutrition from the cell. Mitochondria provides its host with capability of reducing its oxygen content and simultaneously supplying it with a stable form of chemical energy, that is the ATP or adenosine triphosphate. Mitochondria can increase their size, replicate DNA, and undergo fission. So from one mitochondria, we may get two mitochondria. There's replication. Mitochondria also has protein. It can produce its protein. Some of the protein are encoded by the nuclear DNA also. It contains also the RNA, and it contains also DNA, as we discussed. We got all of our mitochondria from our mother. How? Father contributed the spermatozoa. The sperm head will penetrate the ovum for fertilization. Middle piece tail will not. So there is no contribution of mitochondria in our body from our father. We got all the mitochondria from our mother. This has a lot of forensic and anthropologic evolutionary importance because we get all the mitochondria from our mother. Okay. Clinical application, mitochondrial myopathy. As we discussed, mitochondria are present plenty in our skeletal muscle, cardiac muscle. It is present in our epithelial cell of the kidney, present in the liver. Mitochondrial myopathy will have pain in the muscle. We have also weakness of the muscle. We have the Melas syndrome, Marp syndrome, Leigh disease or syndrome. We have a lot of other syndrome. Melas syndrome, that is the mitochondrial encephalopathy, lactic acidosis, stroke-like syndrome, Marp syndrome, myoclonus epilepsy, and rigid red fiber. So we got mitochondria in Gomori stain. It will give red color. Here, this is the skeletal muscle histology, and these are the mitochondria that gives the rigid red appearance in Gomori staining. That is a part of the mitochondrial myopathy, specifically the Marf syndrome. It is very much prominent in this syndrome. We have also Leigh disease or syndrome, that is a subacute necrotizing encephalomyopathy, that is also a mitochondrial disease. We have a lot of other diseases. We may have blindness due to mitochondrial disease. This is a problem. Okay. So what are the highlights of our mitochondrial discussion? Organelle can move from one part of the cell to the other part, like that of the movable power plant. Number of mitochondria varies depending on the function and demand of the energy and also activity of the cell. Location also varies. Outer membrane, inner membrane. Outer membrane is thicker, inner membrane is thinner. Outer membrane is more permeable, inner membrane is less permeable. These are the cristae. shelf-like structure, and we have the ion transport mechanism here. We have the ATP synthetic plant here. Okay, in the matrix, we'll get all the enzymes for the cyclic acid cycle. And citric acid is we'll get from glucose, pyruvate, acetyl coenzyme A, fatty acid will go through beta oxidation and we'll get the acetyl coenzyme A. Then with TCA cycle, we'll get release of carbon dioxide and we'll get the ATP from there. Apoptosis is the programmed cell death, and the cytochrome C that is present in the intermembranous space that is derived from the inner cell membrane that is very essential for apoptosis. It has the capacity for self-replication. From one mitochondria, we may get multiple mitochondria. It is of maternal origin. Okay, so dad has no contribution in our mitochondria. It is from our mother we got all the mitochondria. Okay, so these are my references of my presentation. If you like my video, please let me know, and if anything if like, please let me know also. Please share the information with your friends, and please support my channel. Please subscribe me, and have a nice day. Bye now.